Hello, my name is Sabrina Templo. Today is June 4th, 2020, and this is for Bio 225, week 4 for motility testing. And so basically for this week, we did our motility testing for our E. coli culture and for my S. epidermidis culture. So I'm going to start first with my E. coli culture because this is the one that I actually inoculated first. So when we are inoculating these, we have to make sure that we inoculate them with a stab, or they call it actually a stabbing technique. And we actually did it with a paper clip that was straightened, or a straightened paper clip. So probably in the lab they have more about something better than to stab it with, but basically when I did this, I want to make sure that my paper clip is as straight as I can get it, and when I stab in, that I pull out in the same way, because it will defeat the purpose of keeping the bacteria in one motion and in one spot. So basically what we do is we take our paper clip and it's sterilized with alcohol. You take some cultures from your previous culture, and this is my culture. You can't really see the stab line. It is kind of hard to tell. That you could see that there is turbidity all around. It's kind of like a spider web, or even like a pulled cotton. It is kind of hard to see, but it's easier when you put it against the light. But yeah, there has been a lot of growth, as you could probably see it. But if it had stayed along the stick line, then it would have been negative or non-motile, but there is growth. And that makes sense because E. coli is a bacteria that is motile. And so this would be considered as a positive test. So you want to make sure that there is growth along the stick line, but this has growth all around, as you can see that. I could immediately see the stick line when I inoculated, so I know that I did this one correctly, and I did do this one first. The next one that I did was my S. epidermidis. This one I actually kind of, I was really concerned. These agars were in the fridge, but it was really, I found it was really watery. When I sticked it in, I did not actually see a line, and I didn't know if it was going to grow properly, but luckily bacteria still grew. You could see that there was some, you could see kind of the line, and you can also see that there is some growth on top. I'm not sure that if this was solid as it was supposed to be, even though it was supposed to be soft. I don't know if it was just the weather, because it is summertime. Maybe it was too warm. But when I put the paper clip inside, instead of grabbing around the gel, it probably grabbed more on top as my paper clip left. So, and then it started growing at the top. You could see more turbidity there, but you could also see kind of the line. I'm not sure if this would be considered as a positive or a negative, but I would consider this as a negative because it does have a little bit of a line. I'm not sure, you can't really see it on the video. So I would say my E. coli is positive and my S. epidermidis is more of negative. It was kind of interesting to see the actual motility and growth of each of the E. coli and the S. epidermidis. It was nice to differentiate between both. We can't really see the cells and how they look just by doing these tests. And it was actually interesting to see, and actually I had actually so much fun in doing this, and it was interesting because to see what you can get from your previous plates, and when you put it onto the tubes. And that was really great and awesome to see the growth. And that concludes my results video. Thank you.